Thank you very much. I would like to start with thanking the organizers for inviting me to present our findings today. So I would like to share some data that we collected during the recent years on diaphragm biopsies from critically ill patients. And I'll only give a very brief introduction because this is, I think, known by many of you. So the group of Geert Hermans, of course, but also others have shown that when you stimulate the phrenic nerves with a magnet, that in critically ill patients, diaphragm weakness is observed. This weakness is progressive, as you can see here. On the y-axis, there's time. On the x-axis, there's transdiaphragm pressures. And you can see that this progressively decreases over time. What is not known from these measurements is whether this reduction in transdiaphragm pressure is caused by the phrenic nerve or by neuromuscular transmission failure, by calcium release in the muscle fibers, or by the changes in the contractile apparatus itself, so in the muscle fibers. And I think that, well, based on all the work that has been shown in previous sessions, so these are mainly animal work, where rats are ventilated or mice are ventilated or pigs, that they have shown that ventilation itself can cause huge changes and very rapid changes in diaphragm muscle fibers themselves. And then from 2008 onward, there have been some publications on brain-dead organ donors where they investigated diaphragm biopsies from these subjects. And they also found changes, rapid changes in diaphragm fibers. But it's not known, it was not known if this translates to critically ill patients. So that was our hypothesis, that individual diaphragm fibers from critically ill patients develop a contractile weakness. And obviously, when you want to study this requires biopsies. So several years ago, we initiated a small network, which involved in my hospital the Department of Physiology, in which I am involved, in the ICU, of course, anesthesiology, surgery, and also some external partners, including Leo Hunks, who has been great in advising us here in Nijmegen. We get biopsies from a hospital in Enschede, in the eastern part of the country, another hospital in Amsterdam, and recently also from Ottenheijm, another city in the northern part of the Netherlands. So I will come back to this later. We also collaborate now with a new company in the Bay Area near San Francisco. So we've been collecting diaphragm biopsies, and of course these patients do not undergo surgery just to take a biopsy. So these are critically ill patients that have to undergo a thoracotomy or laparotomy for a clinical reason, of course. And then depending on where, for instance, the laparotomy is, a surgeon can reach the diaphragm and take a very small biopsy. So these are biopsies that are maybe 20 or 30 milligrams, so they are extremely small. Not disadvantages to the patient, but with these biopsies we can do a lot of work. And so you can see this is a summary of, so these are not brain-dead organ donors. I want to emphasize that this is the real deal. These are critically ill patients that are detubated, many of them later, and live on with their lives. So they were ventilated for 30 to 603 hours. It's a very heterogeneous group. Some were septic, some were non-septic. Some use corticosteroids, others less so. It's a very heterogeneous group. And we matched that with a control group, which are obviously not healthy controls, but these were patients with a small primary lung tumor with no other illness that could affect the diaphragm. So they were matched with respect to age, BMI, and also with respect to gender. So we had 22 critically ill patients in this study and 18 control subjects. Another important aspect, I think, is that we aim to rule out criteria or entities that could have caused muscle weakness in these patients before they entered the ICU, such as severe COPD, chronic heart failure, pulmonary hypertension, etc. So we assumed that when these patients entered the ICU, that they had no diaphragm weakness. So biopsies were obtained, as I mentioned before, either through a thoracotomy or through a laparotomy. And then with these biopsies, we either look at protein analysis or muscle fiber structure, or we look at contractile function. I'll briefly explain this a bit later, because it's a bit more specialized technique, but I'll start with the structure. So this is one of the first patients that we analyzed. So this is a diaphragm biopsy, and these fibers are cross-sectioned. So this is a fiber, and this is a fiber. And you can immediately see that in the critically ill patients, these fibers are much smaller. So this patient, if I recall correctly, was ventilated, I think, for 3 days. So this was 3 days after admission to the ICU. And this is what you get. So small fibers, and also a lot of additional nuclei, which turned out later to be inflammatory cells. So there's huge inflammation, whereas this patient had no overt sepsis. Okay, so we quantified this, of course. So we did we fiber typed these fibers. So these are, again, individual muscle fibers. These are cross-sections, so this is the same cell as this one. Here is stained for slow myosin heavy chain, so to identify slow twitch fibers. And this is a stain in green for fast twitch fibers. We have to do this because these fibers each have different characteristics. This is a control patient. This is the same critically ill patient as shown before. You can see that these muscle fibers are much smaller, both in slow twitch and fast twitch fibers. We quantified this. And well, you can see that both in slow twitch and fast twitch muscle fibers there was a marked atrophy. So like a 25% reduction of cross-sectional area. Typically, when a muscle fiber becomes smaller, when they become thinner, they also generate less force. So this could be a strong indication for weakness. So from the same patients, actually this is the same patient as I showed before. This is an H&E staining. It's just a general protein staining. You can see that well, you can maybe debate a bit whether this is a non-respiratory muscle, especially in critically ill patients, but at least it's much less involved in inspiration than the diaphragm and expiration. You can see that we obtained the biopsies from these muscles from these patients. You can see that much less is going on. Also, when you do these fiber type stainings, you can see also per fiber type that not much difference is present yet in these patients. So again, we quantified this. So we found no significant differences. So please understand me correctly. I don't want to insinuate that nothing goes on in peripheral skeletal muscles. I only want to suggest that in the diaphragm, changes might precede the changes that are observed in peripheral or non-respiratory muscles. So this takes me to the second part. We also, in our lab, do a functional study. So we isolate muscle fibers. And actually, I have it better in the next slide. So this is a setup that we use. Now we have 3 of those, so we can do many muscle fibers on a day. And in this setup, first we isolate from the biopsy individual muscle fibers. A muscle fiber is clipped between very small aluminum clips. One side is attached to a length motor, the other side to a force transducer, and then we can expose these fibers to incremental calcium concentrations. So these fibers are not intact anymore, they are demembranated, and that allows calcium to freely enter the cell and activate the contractile apparatus. So we can basically bring back to life muscle fibers that have been obtained from a patient, let's say 10 days before, and they start producing force again. So this is a diaphragm fiber from a control subject, and I hope you can see the nice striation pattern. So this is one individual cell. And this is what you get when you activate them. So this is calcium concentration on the x-axis, force in millinewton on the y-axis. And when you give more calcium, you get this sigmoidal, kind of like a dose-response curve. And at one point, at saturating calcium concentrations, you get a maximal force. So we did this in the slow twitch fibers of the control subjects. You get this relationship. And then in the critically ill patients, we found a more than 50% reduction in the maximal absolute force. So these fibers generate less than half of the force compared to control subjects. And again, so these were ventilated on average 200 hours, so let's say 5 days. So just to give you an impression about the variation between patients, so these are the controls. So this average is the same as this average, and this average should be the same as this average. But here you see the individual numbers. So each dot represents one subject. And you can see that, well, almost all critically ill patients showed severe weakness. We also showed before that these fibers are atrophied, and that contributes to weakness. So what we did next is we took the maximal absolute force and normalize that to the cross-sectional area of each individual muscle fiber that we measured in our setup. And that gives you tension. So in this figure, the factor atrophy is ruled out. It's taken out of the equation. Then still you can see that in these slow twitch fibers, the tension is reduced in critically ill patients compared to control. So this suggests that in addition to atrophy, also intrinsic weakness of these muscle fibers contributes. So the contractile material is probably damaged. In fast twitch fibers, we basically found the same results, although the change in tension was borderline significant. So atrophy and contractile weakness of the individual fibers probably contributes to weakness. So why would tension be reduced? So this is an electron microscopy picture. This is one fiber with a low magnification, 8,000 times. This is a higher magnification. So this is the typical striation pattern in striated muscle fibers. This is a sarcomere. You can see the myosin-based thick filaments. This is the Z-disc. This is a nice structure. You can see mitochondria over here in between the myofibrils. But in the patients, we saw, I must say this is an extreme example, but this is what we saw in many patients. So you can see that, so there's a myofibril here, there's one here, but there's a lot of damaged material in these muscle fibers. So this is a zoom into one muscle fiber. When you zoom in further, you can see some intact myofibrils over here, but also a lot of damaged material. So we think that this actually also contributes to weakness. So in addition to atrophy. So we correlated our maximal force data, absolute force data from these fibers with the hours on mechanical ventilation that these patients were exposed to. And we found these correlations for slow twitch and fast twitch fibers here on the right. You can see in the slow twitch fibers this is significant. So with more hours of ventilation, you get a decrease in force. But I think we have to be very careful with these correlations because this is a very heterogeneous patient group. And this is merely an association. This is far from a causal relation, I think. Probably much more complicated. So my final slides. A therapy that might be interesting is to augment contractile function of these sarcomeres, of these myofibrils in these muscle fibers. And Alexander already touched on this. He didn't mention the potential effect of calcium sensitizers. Here in Nijmegen, they worked on that with Lever Zimondan, and they showed that when you activate troponin in the muscle sarcomere, then you can get a higher force response, and that could potentially help these patients. So we have been in contact with a company in near San Francisco. They're called Cytokinetics, and they provided us with a fast troponin skeletal muscle activator. And this activator works on fast twitch muscle fibers only. And an advantage of that might be that it doesn't affect cardiac tissue because slow troponin isoforms, slow skeletal muscle troponin isoforms are present in skeletal muscle, but also in cardiac tissue. So we use this activator. So what it basically does is it binds to troponin, and as a result, calcium stays longer attached to the troponin complex, and you get a more forceful contraction. So we did a dose-response curve. On the y-axis, the dose. On the x-axis, the dose. On the y-axis, force of an individual diaphragm fiber. We selected 5 micromolars. So this is all in vitro work on isolated diaphragm fibers in the setup that I just showed you. This is again the data that you saw before. So fast-twitch muscle fibers of critically ill patients, they are weak at all calcium concentrations. And then when you expose them to this compound called CK2066260, so this is an analog of tirasemtiv, which is in the phase 2b clinical trial at the moment. You can see that force, especially at physiological calcium concentrations, which are indicated here in yellow, you can see that the light blue line, especially in this area, overlaps with the control line. So this compound is very potent, at least in vitro, in augmenting force and restoring force back to control levels. So maybe in the future this might also be an interesting compound to investigate. So take-home message, diaphragm fibers in these patients develop severe contractile weakness and atrophy, and improving contractile strength might be actually a very effective strategy to help in weaning, with weaning failure. Thank you very much.
Thank you very much for your talk. Are there some questions from the audience? Yeah, so I have one question. Maybe we could imagine that diaphragm weakness is somehow protecting the muscles against like some myotrauma maybe or something like that. If we restore too early the force of the muscles, could there be a risk of generating some trauma? Or this is only speculative, but could you comment on that? Or
yeah, that has not been investigated, but I could speculate that you don't want to do this too early. And that you only want to apply something like that in a more in a phase where underlying disease is maybe diminishing, and you really want to get a patient from a ventilator, and diaphragm weakness has become a main issue. And maybe if you titrate it carefully, slowly increase the dose, and that you can tip them over the hill and get them off the ventilator and breathing on their own. I think that's pure speculation. But I think, yeah, it weakens for a reason. And I can imagine that you don't want to give this at a very early stage, indeed.
Thank you for this excellent talk. I was wondering if you had looked at the difference in mechanical mode, mechanical ventilation mode, if the patients were assisted rather than controlled.
Your question is if there are differences?
In the ventilatory mode.
Yeah, that's why. Yeah, that's one of the heterogeneous aspects. There are huge differences. Some are ventilated, controlled for 60% of the time, and others are ventilated controlled for only 10% of the time. And sometimes that had changed over time. So first we were on full control, then assist, then control again. Sometimes they were off for some time, and then again on. So it's very heterogeneous. So it's very difficult, I think, to stratify this. We will try, especially when we have more patients. We are still including patients, but that will be an important aspect, yeah.
And again, probably very difficult to stratify, but you showed the biopsy of abdominal muscle, which looked still very good. So did you have some patients with general weakness versus other patients with no ICU peripheral muscle weakness?
We only quantified this by means of these biopsies, and some patients, there was quite some variation in these patients. But again, this variation did not very well correlate with clinical entities, like duration of mechanical ventilation. It was very heterogeneous. But yes, some patients had in their so-called peripheral skeletal muscles, or non-respiratory muscles, they had, these fibers were much smaller than in other patients, yes. Okay. But some were also very huge, so that's why on average there was no difference.
Okay. Leo?
Coen, does this troponin activator affect muscle relaxation as well? Because in that case, if there's a prolonged activation, perhaps eccentric injury may be induced. So yeah. Is that a risk, or doesn't it affect relaxation at all?
It does. So they did investigate that, and it does. In our setups, that's hard to do because you have diffusion issues, etc. But they did this on intact muscle, and yes, it does have an effect on relaxation time, but it was minimal. And they also tested it in vivo, and they saw no effects on how these animals ran, or it is not tested in the diaphragm, not on a muscle which is very active, which has a very high duty cycle. So their relaxation effects might be more pronounced or more detrimental than in skeletal muscles that are less active.
But do you agree that it would increase the risk of eccentric muscle injury, or? Because if there's exhalation of the ventilator and the diaphragm is still contracted.
Yeah, that would induce lengthening contractions. You're right. Yeah, true. Yeah.
Okay, great. Thank you very much, and thanks to all the speakers.